Good morning, guys. This is our presentation. My name is Figure Ella, and these are my group mates, Jessica Bergman and Seraphine Wu. This is our article that we worked on. It's called The Evaluation of Possible Molecular Toxicity Induced by Occupational Exposure to Lead and Concomitant Effect of Smoking. It's by Asa, Narmeen, Iman, Fatma, and Mano. So this is the background of in the article, the main thing that is used to perform the experiment is blood, and the reason why blood is used is because it circulates our body with nutrients, with oxygen, and even toxins, which is why it is important for us as humans to get blood tested every year. Agarose gel electrophoresis is used, and we all know that this technique is used to separate DNA fragments based on size, and we use blood to get blood lead level, and MDA level, which is a marker of oxidative stress. So what is lead? Lead is a heavy metal found in earth, which is very toxic and is encountered in our everyday lives, like water and fountains, nail polish, toys, jewelries, or even at work. And what is smoking? Smoking is when a substance is burned and the resulting smoke is inhaled to be tasted or absorbed into the bloodstream. And smoking has a lot of chemicals in it that just puts stress on the lungs and the process of breathing.
So the question of our paper is focused on what is the molecular toxic effect of the exposure to lead and the concomitant of smoking. And the hypothesis of this paper is actually focused on that the exposure to lead and associated with smoking will increase. So on with the blood lead levels, the MDA levels, and DNA damage. So in this paper, we only focus on male workers. So in here, we can see the pie chart that is focused on the male workers. So in this experiment, we have two control group. One control group that is the male that is not exposed to lead and not smoke, and another control group that is not exposed to lead but they smoke. And also they have two testing group, that one of them that exposed to lead but not smoke, and the another testing group, they are exposed to lead but they are heavy smoker. So here you can see the comparison between the techniques from the paper and from our class. In this paper, we have learned the agarose of electrophoresis and the blood test. But from our class, we only learned the agarose gel of gel electrophoresis. So because we only learned gel electrophoresis, so the following figures that I'm gonna show you guys is only specific on gel electrophoresis. So in figure 1, this is the result of the control groups that is not exposed to lead. So in here you can see the M is a ladder. The C1 to C6 is the male results. So in here you can see that the DNA bending is definite and the DNA is not fragmented and they are very close to the well. In figure 2, in the figure 2, we're looking on the testing group of the lead exposed worker. So same thing, the M is still the ladder. The E1 to E7, they are the male workers. But in here, you can see that the bending is no longer definite, and the DNA are more fragmented. Then they are like in a mild fragmentation. Now in figure 3, you can see like a big comparison to figure 1 and 2. So this one is the testing group from the heavy smoker. So in here you can see that the DNA is more fragmented compared to figure 2 because they are the heavy smokers.
Now, isn't that crazy? So essentially what happened? So the first thing was researchers obtained a sample of subjects and put into 4 groups. Lead exposed non-smokers, lead exposed smokers, non-exposed smokers, and just not exposed to anything. What did they do with these people? They took blood. From this blood, they obtained the blood lead level, the MDA levels, which are a marker of oxidative stress, and gel electrophoresis. And what did we result in? Well, you just saw the gel electrophoresis, and what did we see? Insane fragmenting. Just look at what happened to the lead exposed workers and smokers. That was insane. You would never want that done to your body, and that's why don't smoke. No, but the conclusion that can truly be drawn from the experiment is that, based on the data that the researchers collected, it does support the hypothesis, and it did depict a very strong correlation between exposure and toxic effects. As exposure increased, blood lead level increased, MDA level increased, and fragmentation would occur. So in other words, what happens to blood lead level? Blood lead level were increased for workers exposed to lead. That makes sense. Exposure to lead equals more lead in the blood. Plasma malondialdehyde levels, the MDA levels, are increased for those exposed to lead and smoked. That makes sense. They're a marker of oxidative stress. When you have all these toxins, it's hard to breathe. Gel electrophoresis, that was really interesting because as this toxin entered your body, it altered the length of your DNA. It damaged your DNA by breaking it up. That's really, really interesting. So now the key findings, what were they? So the researchers answered their question based on the constricted experiment. They asked what was the effects of it, and they found out, but it was constricted to a small sample of 60 men between the ages of 33 to 61. So we can't really say this applies to everyone. Also, their hypothesis was supported by the experimental results in the data. And as exposure to lead and smoke increased, so did blood lead levels, plasma malondialdehyde levels, and DNA fragmentation. Now, what was the whole point of the study? Why would they do this? What benefit does it bring to them? Well, there's more information on this, more toxic effects of lead and smoking that needs to be spread. People need to know this in order to caution workers and protect people for the future. You don't want the whole world of workers dying because they're just not aware of what's going on in the work environment. But in order to do this, we need some follow-up experiments. We need to do an experiment to help prevent more damage and protect lead exposed workers. We need to do an experiment that's broader, that involves more people, that involves more correlation, more smoking tests, more analysis of what is going on in order to create an actual correlation and statement on what's going on. So just some wrapping up, and just to get this very clear, 100% of the people in the study were males. Not one person was a female. The experimental time, it was done within a year. That's pretty normal. The results, everything was increased from the exposure. So just to wrap up, what's our conclusion? Our conclusion is literally that lead and smoking are 2 very damaging toxins, and when co-exposed, blood lead levels increase and oxidative stress is put on the body and DNA fragmentation occurs. And while our conclusions are supported by the experiment, more research needs to be done on more people to depict a true trend. However, they did perform a very well planned experiment, and they did obtain results that are very, very viable, and we can use this to spread information on the dangerous substance we encounter on our everyday lives. So these are our references. We even have the CDC here, as we all love, and our article. And now is the time for you to wrap up and ask all of your questions. So please, this is the space to do so. If you're watching this on YouTube or watching it at home, you can shoot us an email or write in the comments below. And we flashed to the ends. Thank you for listening. And we're done.